Now we are going to see the various stages which are involved in the process of recombinant DNA technology. We had seen the various tools those were involved. Now we are going to see what is the utilization of those tools, how those tools are utilized. Now before we start with the stages, this is our topic that we are going to cover in this lesson, the stages which are involved in the process of rDNA technology. Before we start with the stages, we have to see the two main characters which are going to be involved in this process. Like we have main characters in a movie or in a play or in a story, we have two main characters over here as well. The first one is that we should have in the end is the transformant, and the second one is the recombinant. These are the two main characters of our story that we have. In various stages it would be carried out. Now what is a transformant? Transformant is the host. It is the host cell inside which the recombinant is incorporated. Now what is recombinant? Recombinant is that vector. You remember what a vector was? Vector, we usually had the example of plasmid and we had discussed it, that what is the purpose of vector, why do we have vector in this rDNA technology. The plasmid which has the gene of interest incorporated inside it is known as the recombinant. When the plasmid has recombinant DNA inside it, it becomes the recombinant. Now this recombinant has to be incorporated into the host cell. This recombinant would be introduced into the host cell and that would transform the genetic composition of the host cell. Then the host cell becomes transformant. So with the help of a recombinant vector, we are going to achieve a transformant, and we are going to see what all stages are involved in this process. Now step by step we will see that first of all we need an alien DNA. First thing is that you need a gene of interest that would be taken from some genome. Okay, you need a vector that would be a plasmid, and you need a host cell, which in this case we have E. coli. One by one we will see what we are talking about. Now supposedly this is your gene of interest, okay, that you have taken from some particular organism, and we are going to see individually how this is extracted, because we deal with the entire methodology when we study it. So for once you just have to see that a gene of interest has to be taken from a genome by using restriction endonucleases, then it has to be incorporated into the plasmid. Supposedly this is the plasmid. Now I have to be very particular in drawing. This wavy pattern, I have tried to represent the DNA, though it does not look like that. Now this gene of interest, using the same restriction endonucleases, will be incorporated into the plasmid like this. Okay. Now a plasmid that has the alien gene inside it, as you see the composition of plasmid has changed, the alien gene has got incorporated over here, and it is known as recombinant. Okay. How this process has been carried out? It is carried out using restriction endonucleases, right. Now this restriction endonucleases will cut it and form the recombinant DNA that we otherwise call as Rdna vector. This is the vector because it is a plasmid. Now it will be incorporated into E. coli cell. Supposedly E. coli is a prokaryote, you know that. Now This is its nucleoid. Here we have the plasmid that is being put inside the E. coli, and this plasmid is not the simple one, it is the transformed plasmid. Now we have this plasmid. This E. coli earlier did not have this plasmid because of the process of transformation. Step number 1 is recombination. And step number 2 is transformation, where we have the vector getting incorporated into the host cell, that happens to be E. coli. This is E. coli. We have its own nucleoid and the recombinant that has been introduced, and this E. coli with external DNA or the recombinant DNA is known as transformant. So, this is what is the entire process, the various stages which are involved in the process of recombinant DNA technology. The first step happens to be of recombination and the second step is of transformation. In the first step, we use restriction endonucleases, cut the gene of interest from the given genome wherever it is found, then incorporate that gene of interest into the vector using certain digestive enzymes, not digestive, pardon me, there would be binding enzymes, those are known as ligases. We have studied about it in genetics. We have ligases. These ligases join this alien DNA piece into the vector. Now this vector, using certain enzymes and certain methodologies, again I would say, is incorporated into the host cell, which earlier is not having a plasmid, and that leads to the process of transformation, resulting in formation of a transformant. So these are the various stages. Simplest presentation, where we have the resultant transformants being formed as a result of rDNA that resulted into recombination. The vector was a recombinant DNA genetic entity, and that got incorporated into the host, and that was a transformant. So these are the various stages in rDNA technology that you have to keep in mind, and we have lessons where we dedicate a good amount of time in understanding what all methodologies at the laboratory level would be required for carrying out these processes.